Imagine this, the tiny device on your wrist, maybe the smartphone in your pocket, things that maybe start out as just lifestyle gadgets. They're quietly transforming, becoming these really powerful medical instruments. They're collecting data, not just your steps, but like intricate physiological signals right there in your actual life. So what if that everyday smartwatch could fundamentally change how we develop new medicines, how we run clinical trials, and how we understand and treat diseases? Welcome to the Deep Dive. This is where we take a plunge into, well, a fascinating stack of sources, articles, cutting-edge research, and together we pull out the most important nuggets of knowledge, the key insights. Today, we're diving headfirst into exactly that question. Our sources, they really explore the incredible and pretty complex regulatory journey of what are called digital endpoints. These are those crucial, precise measurements derived from data captured by wearables, other digital health technologies, DHTs, you'll hear us call them, within the very rigorous world of clinical trials. So our mission for this deep dive together is basically to cut through the noise. We'll unpack the really rigorous validation processes behind these digital tools. We'll celebrate a truly landmark regulatory success that's kind of paving the way. And we'll try and grasp the critical challenges that are shaping this new frontier. Consider this your fast track, your shortcut to understanding one of the most transformative shifts happening in modern medicine right now. Okay, so on this journey, first we'll uncover what digital endpoints really are, how they differ. Then we'll look at a groundbreaking regulatory approval from Europe that's really set a precedent, a big one. From there, we'll meticulously dissect the gold standard framework used to validate these measures. And then, we'll tackle the hurdles. Things like managing messy real-world data, or even the sort of intriguing, sometimes complicated idea of using your own personal device in a trial. Okay, let's unpack this.
Yeah, it really is a pivotal moment, I think, in how we measure health. For decades, clinical trials have mostly relied on periodic, sometimes quite artificial, snapshots, right? A patient's health measured during brief clinic visits.
Right, like once every few months, maybe?
Exactly. Think about it. A single measurement, maybe every 3-6 months. But with these digital health technologies, these DHTs, we're seeing a huge paradigm shift. We're moving towards continuous real-world data collection. And that provides just an unprecedented richness, a depth of information about someone's condition that we just haven't had before. It's like moving from a few still photographs to a continuous high-definition video of someone's health journey.
A high-def video, I like that, #DAG-I. The rise of digital health technologies, DHTs, and digital endpoints. And that continuous video of data, that's exactly where we need to start, isn't it? What are these digital health technologies, these DHTs that are making all this possible? So when we talk about DHTs, we're essentially looking at a pretty broad category, right? Devices, platforms that capture health-related data, but crucially, outside of that traditional controlled clinic setting, it's really about leveraging tech for health monitoring in everyday life. So this includes, the wearables you might already on your smartwatch, fitness tracker continuously tracking steps, heart rate, sleep, all that stuff. But it also goes further. It includes smartphones, which are basically, mini computers packed with sensors. They can measure everything from your activity levels, even voice patterns for certain conditions, believe it or not.
That's right.
And beyond that, it encompasses more specialized passive sensors, things that can be integrated into a person's environment, maybe pressure mats under a bed, tracking sleep, or smart home devices monitoring movement, maybe even environmental factors relevant to health.
And the range is quite staggering when you think about it.
It really is. And the truly incredible capability here, I think, is their ability to give us these continuous real-world measurements. They're not just taking one reading at one specific moment. They're constantly monitoring, observing, collecting data throughout a person's day, sometimes for weeks, even months on end. We're talking about everything from basic step counts seems simple to highly sophisticated gait analysis detecting really subtle changes in walking patterns. They can track the frequency, the severity of tremors, monitor fluctuations in heart rate, analyze sleep architecture, gauge overall activity levels. It's this constant stream of really granular information collected in someone's actual natural environment.
Exactly.
And that just paints a far more comprehensive, probably more accurate picture of a person's health than those traditional episodic clinic visits ever could. It really is the difference, like you said, between a still picture versus a dynamic film of someone's daily life.
Precisely. And that continuous stream, that high definition video, as you put it, leads us straight to the concept of digital endpoints.
Yeah.
Because, it's not enough to just collect all this raw data.
Right.
It needs to be processed, analyzed, refined into meaningful, quantifiable measures, things we can actually use effectively in a clinical trial to see if the treatment is working. And what's really fascinating here, I think, is that these digital outputs, they exist along a kind of spectrum, a continuum, based on their intended purpose and what they're actually designed to measure.
Okay, a spectrum. How does that work?
Well, on one end, you have what we call digital biomarkers. Think of these as physiological surrogates. They might measure a specific biological or physiological process that tells us something about the disease itself, or maybe how a drug is impacting a particular mechanism in the body.
So like an internal indicator.
Exactly. Objective, often subtle indicators of biological change. Then further along this spectrum, you find digital clinical outcome assessments, or COAs. Now, these are measures that relate much more directly to how a patient feels, functions, or survives in their daily life. They have direct functional or symptomatic relevance. They tell us something we can observe, something meaningful about the patient's actual experience.
Okay, so biomarker is more internal mechanism. COA is more about daily life function.
Pretty much. And then, importantly, there are hybrid endpoints sitting right in the middle. These are really interesting because a single metric might actually serve both purposes. It might be designed to provide evidence for both a physiological impact and real-world functional relevance. Now, the crucial point to grasp here, and this is absolutely vital for anyone in drug development regulatory science, is that the regulatory path depends entirely on the intended use of that endpoint.
Right. It's not just what it measures, but why you're measuring it.
Precisely. It's not just about what the device can measure, but what you want it to prove in the context of the trial. Let me give you an example, maybe make it clearer. Think about something seemingly simple, like walking speed.
Okay.
If you're studying a drug for, say, a neurological condition, one that affects specific motor pathways in the brain, and you use walking speed to understand how the drug is impacting that precise neurological process, maybe improving nerve signal transmission, that it's functioning as a biomarker.
Got it. Physiological surrogate.
Exactly. But if you're assessing a drug for mobility impairment, like in Duchenne muscular dystrophy, and walking speed is being used to measure a patient's functional ability to just get around in their daily life, how quickly, how effectively they move through their home or community, then it becomes a clinical outcome assessment, a COA.
Because it's measuring their actual function?
Yes, a direct measure of their functional ability in the real world.
Yeah.
And this distinction, biomarker versus COA, it's absolutely critical. It dictates the type, the rigor of evidence that drug developers need to present to regulators like the FDA or the EMA.
So it changes what proof they need.
Completely. If it's proposed as a biomarker, you need robust data showing it accurately reflects that specific biological process. If it's a COA, you need to prove it reflects something meaningful to the patient's daily life, that it's clinically relevant. Often that involves patient input, comparing it to established clinical scales. This difference, it impacts everything. The study design, the statistical analyses, the patient population you choose, and ultimately whether a drug can get approved based on that measurement. It defines the whole evidentiary burden.
So, okay, what does this all mean for you, the listener, maybe the patient, or just thinking about the future of treatments? It sounds like digital endpoints mean potentially less guesswork, right? Imagine your doctor maybe not just asking, so how's your walking been during a quick clinic visit? Instead, a digital endpoint could objectively show your peak walking speed over weeks, providing real data about how you're actually functioning day to day.
That's the goal.
We're shifting from these subjective snapshot assessments towards objective continuous proof of a drug's real impact on your life. It allows for a much more nuanced, accurate, and truly relevant understanding of how well treatments work. It's pushing medicine closer to being really precise, tailored to individual needs, and this shift towards objective continuous measurement, it brings us perfectly to a real-world example, a really big deal in the regulatory world, the qualification of something called SV95C.
Ah, yes, SV95C.
No, SV95C. It stands for Stride Velocity 95th Centile. It's a bit of a mouthful, yeah, but it's a game changer because it measures something incredibly specific and relevant, a person's peak ambulatory capacity. Basically, it tracks your maximum comfortable walking speed. But here's the really crucial part. It's captured in your real-world daily conditions. Not on a treadmill, not in some sterile clinic hallway.
Right, in their natural environment.
Exactly. And how's it derived? It comes from continuous data collected by a small wearable device worn on the ankle. And we're talking a significant amount of data, typically around 180 hours of monitoring, collected over several weeks.
Yeah, it's not a quick snapshot.
Definitely not. It's a dynamic, longitudinal measure of how someone moves in their own environment, doing their everyday things. This is just profoundly more reflective of their actual experience, their functional ability, than any traditional in-clinic test could ever be. It captures all that natural variability, the challenges of real life.
And the regulatory significance of SV95C, well, it really can't be overstated. This isn't just some interesting academic finding. It is the very first digital primary efficacy endpoint that has been qualified by the European Medicines Agency, the EMA.
The first primary digital endpoint. Wow.
The first. A major milestone achieved in July 2023. And specifically, it was qualified for use in Duchenne muscular dystrophy, DMD, which is a severe progressive muscle wasting condition.
Okay, so why does that matter so much? Why is primary efficacy endpoint such a big deal here?
Because this qualification, it powerfully demonstrates a clear regulatory willingness, a signal that agencies like the EMA are ready to accept wearable-derived primary efficacy endpoints, provided, and this is key, they are rigorously and properly validated. What's truly groundbreaking here is that designation, primary efficacy endpoint. To put that in perspective for the industry, for anyone listening, the primary efficacy endpoint is the main outcome measure in a clinical trial. It's the one that ultimately determines whether a drug is considered effective enough to be approved and sold.
So it's the main hurdle the drug has to clear.
It's the main hurdle. It's the central piece of evidence that a drug's fate really hinges on. Until now, using wearable data for that specific purpose, well, it's often been viewed with, let's say, considerable skepticism.
Right.
Or maybe it was relegated to secondary analyses, exploratory stuff, seen as supplementary, maybe interesting, but not definitive for approval.
So this changes that perception.
Massively. This qualification by the EMA provides a critical precedent. It offers a clear pathway. It sends an incredibly strong signal to pharma companies, to researchers, to tech developers everywhere, that these digital measures are truly viable for making drug approval decisions. It validates the huge investment, the effort required to develop and validate these technologies. And this effectively, you could say, opens the floodgates for other digital endpoints to follow a similar rigorous path.
Yeah.
It could revolutionize how we assess treatments across a whole range of diseases, not just DMD, and ultimately speed up getting new therapies to patients.
The implications are, truly immense. It paves the way for much more objective, continuous, and ecologically valid data. That's the term, right? Data collected in the real world, not some artificial setting.
Exactly. Ecologically valid.
Think about the limits of traditional tests, like a 6-minute walk test in a clinic. Okay, it might show a patient can walk X distance on a flat, controlled surface. But it doesn't tell us how they manage stairs at home, or uneven pavement outside, or how their fatigue fluctuates through the actual day. SV95C, though, it gives us data about their peak walking capacity in their actual life, navigating all those real-world variables.
That's the key difference.
So this objective, continuous data can start to maybe supersede those traditional assessments that only capture a fleeting snapshot. It's fundamentally more insightful, more reliable as a measure of how a disease impacts daily function, and crucially, how a drug might truly improve it. It offers a level of precision, that real-world relevance we just haven't had before. It shifts the focus from can they do it for 6 minutes in the lab to how do they actually live their life day to day.
That's a great way to put it.
Okay, but how do you actually convince regulators that these measurements from a little device on an ankle are truly reliable? Reliable enough to be the gold standard for efficacy, especially for that critical primary endpoint. That sounds like the million-dollar question, maybe billion-dollar question.
It certainly is. And the answer, or at least a major part of it, lies in a really meticulously crafted framework. Our sources tell us the EMA's qualification of SV95C relied heavily on what's known as the V3 analytical framework.
V3.
Yeah, it's basically a three-pronged approach. You could think of it as the emerging gold standard for validating these digital measures. It's becoming the blueprint for regulatory success in this space.
3 prongs, what are they?
Right. So the V3 framework is designed to ensure that a digital endpoint isn't just, statistically significant, but that it's truly accurate, reliable, and clinically meaningful. The first stage, V1, is verification.
Verification.
This focuses on the technical side of the device itself, its ability to capture data consistently. It's about making sure the device is properly calibrated, that its raw measurements are precise, repeatable. If we connect this to the bigger picture, This is the foundation, right? You absolutely cannot build a robust, trustworthy endpoint on faulty or inconsistent measurements from the hardware itself.
Right, garbage in, garbage out.
Exactly. It's like trying to build a skyscraper on a shaky foundation. It just won't hold up under scrutiny. So this stage confirms the wearable is doing what it claims, accurately and consistently capturing that raw data acceleration, heart rate, whatever it is.
Okay, so V1 is about trusting the device's raw output. What's V2?
V2 is analytical validation. This is where you take that verified device data and you rigorously assess its accuracy against established gold standards.
Ah, okay, the comparison step.
Precisely. For SV95C, this meant comparing its real-world measurements against highly precise methods, like lab-based motion capture systems, the ones with specialized cameras, markers, getting incredibly accurate movement data in a controlled setting.
Right, the high-tech lab stuff.
Exactly. And the results for SV95C here were really exceptional. The interclass correlation, or ICC, that's a statistical measure of agreement between two different measurements, it exceeded 0.97.
Wow, 0.97, that sounds really high. What does that mean practically?
It's incredibly high. An ICC of 1.0 would mean perfect agreement, so 0.97 indicates near perfect reliability and accuracy. And furthermore, the measurement error was really low, less than 2.5%. So what an ICC of 0.97 signifies, especially when you think about reliability over months for comparing measurements across different time periods, it's truly remarkable. It means if the device measures a certain walking speed today and then measures it again in a month under similar conditions, you can be extremely confident that any difference you see is a real change in the person's walking ability, not just random device error or noise.
So you can trust the changes it shows over time.
You can really trust them. These numbers demonstrate that the digital measurement from that ankle wearable, collected out in the dynamic, unpredictable real world, is nearly as good and maybe in some ways even better than the perfect lab setting, because it captures that real-world data with almost lab-level precision. This rigorous level of analytical validation is what gives regulators the confidence they need in the data's integrity, its ability to reliably track changes over time.
Okay, so V1 is device verification. V2 is analytical validation against a gold standard. What's V3?
V3 is clinical validation. And this is arguably the most crucial stage. This is where you demonstrate that the digital endpoint has a meaningful association with traditional, well-established clinical outcome assessments.
So linking it back to existing tests.
Exactly. It's about proving that what your device measures actually correlates with what clinicians and patients already consider important and meaningful. For SV95C, this involves showing its strong correlation with existing, commonly used clinical tests like the 6-minute walk test, the 6MWT, and the North Star Ambulatory Assessment, or NSAA. These are standard tests used to assess functional mobility and disease progression in conditions like Duchenne.
Right, the ones doctors already use.
But the key finding for SV95C during this stage was absolutely groundbreaking. It wasn't just that it correlated, it actually detected significant functional change in patients much earlier than the conventional tests.
Earlier? How much earlier?
Significantly earlier. Specifically, it detected change in just 3 months, compared to maybe 9 to 12 months for the 6MWT.
Wow, 3 months versus 9 to 12? That's a huge difference.
It's a massive difference. And this raises a really important question. What does earlier detection truly mean for patients and for drug development?
Yeah, what are the implications?
Well, for patients, it could mean knowing much, much sooner if a treatment is actually working, allowing for quicker adjustments to their care plan, maybe faster access to therapies that could be life-changing, preventing unnecessary suffering or continued decline. And for drug developers, it can significantly accelerate decision-making in trials, maybe allowing for quicker go-no-go decisions, enabling adaptive trial designs, or even stopping a trial early if a drug clearly isn't working. That saves enormous resources and ultimately gets effective treatments to patients faster. It basically compresses the whole drug development timeline.
That earlier detection capability is truly powerful. I mean, imagine the impact, the hope of knowing sooner if a treatment is really working for someone with a progressive disease. Instead of waiting like nearly a year to see a measurable change with a traditional test, a digital endpoint tells you in just a few months. But I can imagine some skepticism too. Could detecting change sooner be problematic? Could it lead to maybe premature conclusions or even, false hope if an initial change isn't sustained long term? What safeguards are there to make sure faster doesn't mean less reliable?
That's a really astute question, and it's a valid concern. The key to mitigating that risk lies, I think, in the robustness of the V3 framework itself, particularly those analytical and clinical validation stages, and then the next step, which is defining meaningful change. Regulators don't just look for any change. They look for sustained and meaningful change. So while a signal might appear early, the validation requires demonstrating that this early signal consistently predicts a meaningful outcome that aligns with established clinical understanding.
Okay, so it's not just the first blip on the radar.
Exactly. It often involves looking at multiple time points, not just the very first detection, and using sophisticated statistical methods to confirm the durability and the clinical relevance of that change. It's really about ensuring the early signal is a true indicator, not just some transient fluctuation. The faster signal helps make quicker decisions, yes, but those decisions are always backed by rigorous data demonstrating a real lasting effect.
That makes perfect sense. So it kind of changes the timeline of hope, as you said, for patients and families, but with that necessary scientific rigor built in to ensure that hope is actually well-founded. So, okay, we've established these digital endpoints can be incredibly precise, reliable, they can even detect change earlier. But here's the next big hurdle, and it feels like a deeply human one. It's one thing to measure a change digitally, right? To say someone's walking speed changed by, 0.15 meters per second. It's an entirely different challenge and just as critical to figure out if that change is actually meaningful to a patient, to their daily life, or to a clinician making treatment decisions. A statistically significant change isn't always clinically significant, is it? How do you bridge that gap between the numbers on a screen and the real-world impact?
You've hit on a really crucial point. This is where something called anchor-based methods for deriving a meaningful change threshold, or MCT, come into play. You're absolutely right. It's not enough to just show a number changed. You have to show that change matters.
Anchor-based methods. Okay, what does that involve?
The approach here involves anchoring the changes you see in the digital endpoint to something that is already understood and accepted as meaningful. So for SV95C, this meant meticulously comparing the changes seen in that digital measure to what was observed in traditional, well-understood COAs, like the 6-minute walk test we mentioned. And crucially, comparing it to what patients themselves reported about their own functional impacts. This often involves using patient-reported outcome measures, PROs, surveys, or qualitative interviews, where patients describe how their condition affects their daily activities.
So you're tying the new digital measure to existing scales and to direct patient feedback.
Exactly. Both are critical anchors. So for SV95C, through this meticulous anchoring process, the meaningful change threshold was estimated to be a decline of somewhere between -0.10 and -0.2 meters per second.
Okay, a specific range.
Yes, and this specific range reflects a level of functional decline that wasn't just statistically detectable by the device, but was genuinely considered meaningful by both patients living with Duchenne and the expert clinicians who treat them. It's about translating that highly technical measurement into a real-world impact that resonates. For a patient, this might translate to questions like, does this change mean I can now walk to the mailbox without stopping? Or am I struggling more to keep up with my friends? It's about bridging that gap between the cold, hard data and the warm, lived human experience.
Making the numbers relatable.
Precisely. And it's also important to note, this wasn't just a single method used in isolation. It was a process of triangulation, bringing together multiple lines of evidence to strengthen the conclusion. Yeah, so the anchor-based methods were combined with other analytical techniques, like distribution-based methods. These look at the statistical properties of the scores themselves, things like the standard deviation, SD, of the scores, the standard error of measurement, SEM, which tells you about the precision of the measurement, and the minimum detectable change, MDC, which is the smallest change that can be considered real beyond measurement error.
So combining different statistical approaches.
Exactly. Distribution-based methods look at the variability of scores within a population to estimate what constitutes a detectable change statistically. By combining these anchor-based and distribution-based approaches, you get a much more comprehensive view. And you really strengthen the confidence that the derived threshold, that -0.1 to -0.20 meters, truly represents a change that is both clinically relevant and personally impactful for patients. This multifaceted approach provides a much more robust argument for the clinical meaningfulness of the digital endpoint.
Okay, that makes sense. You're building a stronger case from multiple angles.
You are. And this nuanced process really highlights the inherent complexity, doesn't it? Deriving clinical significance from purely quantitative data. It's not just about hitting statistical significance, p-values in a trial. It's about making absolutely sure that the observed changes in these digital measurements directly translate to a palpable, undeniable difference in a patient's quality of life or their functional ability. This blending of objective data with subjective experience anchored in clinical relevance is what makes these endpoints truly powerful and genuinely patient-centric.
So this ensures that these high-tech measurements aren't just, numbers floating on a screen or impressive stats in a research paper. They actually reflect what matters most to you, the individual experiencing a condition. It's about taking those precise, continuous streams of data and asking the crucial question. What does this really mean for someone's ability to live their life? Are they able to do more? Are they experiencing less pain, less fatigue? This is where the technology becomes profoundly patient-centric, ensuring that scientific progress genuinely improves human lives. Okay, as groundbreaking as all this sounds, the validation, the meaningful change, these advancements don't come without their challenges, right? There must be hurdles. And one of the initial and surprisingly persistent challenges, even before you get to the really complex data analysis, is simply language, the words we use. We're talking about the problem of terminology harmonization.
Yes. Terminology, it sounds dry, but it's so important.
It sounds like a bureaucratic detail, maybe, but our sources suggest it can have enormous ramifications. They highlight that the rapid development in this field has kind of suffered from inconsistent use of terms across different groups. Researchers, pharma companies, regulators, patient advocacy groups, everyone. For example, people might use digital measure interchangeably with digital endpoint, or confuse EEOA, which is electronic clinical outcome assessment, often data entered by patients on a tablet with a digital COA derived from a sensor. Or even mix up raw data straight from a device with processed features that have already had some analysis applied.
That happens all the time.
And this might sound like, minor semantic quibbles, but in the highly regulated, super high-stakes world of clinical trials, precision in language is absolutely paramount, isn't it?
It really is. What's actually fascinating here is how simple miscommunication, just using different terms for the same thing, can significantly, genuinely derail regulatory review.
Derail it.
Well, imagine trying to build a complex product, like a new drug, worth millions, potentially billions, involving multiple global teams, the sponsor company, the payers, regulators in different countries, contract research organizations, CROs doing the work, and everyone's using slightly different blueprints or different names for the same critical part.
Chaos.
Exactly. It leads to confusion. It necessitates endless clarification rounds back and forth. It causes significant delays in the review process. And in the worst case, it can even compromise the entire integrity of a regulatory submission.
Wow. Just from using the wrong words.
Just from lack of clarity. So to solve this critical issue, organizations like EFPIA, the European Federation of Pharmaceutical Industries and Associations, have proactively developed harmonized lexicons, glossaries. These documents define terms clearly, consistently, ensuring everyone involved in this digital endpoint ecosystem is literally speaking the same language. And that's absolutely essential for smooth, efficient, and defensible regulatory pathways. Without it, you're just creating unnecessary friction and risk.
That's a powerful point about terminology. It really does sound like a seemingly minor detail that could cost millions, maybe years, in drug development. Beyond just publishing an official lexicon, though, what's been the biggest practical challenge in getting everyone on the same page? From the research lab to the regulator's desk, when there are so many different perspectives, established practices.
That's a great question. I think the biggest practical challenge is often just cultural inertia. People get used to their own terms, their own definitions within their own teams or organizations. Changing that requires a really concerted effort. It's about education, collaboration across the entire ecosystem.
So it's more than just a document.
Oh, much more. It's not just publishing a glossary and hoping people read it. It's about active engagement workshops, pilot programs, continuous dialogue between industry, academia, regulators, patient groups to ensure widespread adoption and genuine understanding. The initial effort is high, yeah, but the long-term gains in efficiency, clarity, and ultimately speed are immense.
Right. Overcoming that this is how we've always done it mindset.
Exactly. And beyond terminology, another persistent and really substantial hurdle in these DHT studies is missing data.
Ah, yes, the gaps.
Think about it. When you're collecting data continuously, 204/7 in the real world, well, Life happens. Things go wrong. Devices can be removed by participants, maybe for comfort, showering, activities where they might get wet.
Sure.
There could be software glitches, hardware failures, or even just passive non-adherence, where someone simply forgets to wear their device for a while or lets the battery run out.
Speaking of compliance, I have to admit, I'm guilty of this myself. My smartwatch often ends up on the charger for like half the day, or I forget it entirely when I dash out the door. It makes me wonder, how do scientists even begin to account for potentially millions of people doing exactly that in a large trial? Do they just have to toss out all that incomplete data, or is there some kind of scientific magic involved?
That's a perfectly relatable scenario. And no, we certainly try not to just toss out valuable data if we can help it. That would be incredibly wasteful and could bias the results. Fortunately, there are increasingly sophisticated statistical and data science solutions being developed to address this missingness. Often we refer to these as imputation techniques, basically, methods for filling in the gaps intelligently.
Imputation. Okay.
So for instance, for smaller short gaps, something called epoch-level imputation might be used. This looks at the data immediately before and after the gap, maybe within a short time window or epoch, and makes an informed guess to fill it in. But for longer or more complex patterns of missingness, more advanced techniques are needed. Functional data analysis, fPCA, is one approach. Instead of looking at individual data points, it models the overall shape or trajectory of the data curve over time.
The shape of the data.
Wow. Yeah, it allows the method to robustly fill in missing sections by understanding the typical patterns and flow of movement or heart rate or whatever signal it is, even with some points missing. It's less sensitive to individual missing values because it sees the bigger picture.
And you mentioned machine learning earlier.
Absolutely. We're leveraging the power of modern machine learning, particularly deep learning techniques, things like LSTM-based sequence prediction models. That stands for long short-term memory networks.
Okay, LSTM.
These are being explored quite actively. They're designed to understand sequences in context, much like predicting the next word in a sentence you're typing. They can learn complex patterns from large, complete data sets and then use that knowledge to make highly informed predictions about what data is likely missing in incomplete sequences. It's quite cutting-edge stuff.
Wow, so using AI to fill the gaps.
In a sense, yes. But beyond just filling gaps, it's absolutely crucial to perform what are called sensitivity analyses. This is about understanding the potential impact of different assumptions about why the data is missing. This is particularly vital for distinguishing between data that's missing at random, MAR, versus data that's missing not at random, MNAR. This distinction is critical.
Okay, MAR versus MNAR, what's the difference?
Let's make it clear. Imagine someone takes off their smartwatch because it's uncomfortable to sleep in or they're swimming. That's likely missing at random or MAR. The fact that the data is missing isn't directly linked to their underlying health condition itself. It's related to other factors like convenience or activity.
Got it.
But what if they take it off because their tremor is so severe they can't manage the clasp, or the device is actually interfering with their movement? Or perhaps their pain is so intense they simply forget to put it on, or they're too fatigued to bother. That's missing, not at random MNAR. The reason the data is missing is directly related to the very thing we're trying to measure or the underlying health condition.
Ah, okay. That sounds much trickier.
MNAR is a much, much trickier problem because it can seriously bias the results if you don't account for it properly. If people with more severe symptoms are systematically missing data, your analysis might underestimate the severity or overestimate the treatment effect.
Right.
So our statistical analysis plans the SAPs for clinical trials. They have to explicitly anticipate both MAR and MNAR scenarios. They need to pre-specify how missing data will be handled, which imputation methods will be used, and crucially, include sensitivity analyses to check if the main conclusions hold up, even if the missingness is MNAR. It's about ensuring the conclusions drawn are valid, robust, and defensible, even with these real-world imperfections and data gaps. It's about building in resilience against bias.
This is really where the rubber meets the road, isn't it? It's clearly not just about collecting the data. It's about making absolutely sure that data is usable, robust, reliable enough for the incredibly high stakes of drug development and patient safety. It's quite a testament, actually, how adaptable and innovative the field has become, blending this cutting-edge data science with the often messy realities of human behavior and real life. And for all this talk about technology, validation frameworks, data science, it's so vital to remember that at the absolute heart of clinical trials are the patients, real people living with real conditions. And the success of these digital endpoints, it isn't just about technical validation, is it? It's also profoundly about patient acceptance and getting broader stakeholder alignment across the whole healthcare ecosystem.
Absolutely. The human element is paramount.
Our sources highlight some really important findings here from quite extensive patient surveys and qualitative studies. What seems consistently clear is a remarkably high level of acceptance, and often actually a strong preference for wearables among patients.
That's often the case, yes.
People seem generally comfortable, often quite enthusiastic about using these devices, especially if they see a direct benefit, maybe in understanding their own health better, managing their condition, or feeling like they're actively contributing to vital medical research.
That sense of contribution is powerful.
And these studies, they've also been really instrumental in helping identify the specific symptom domains that patients themselves find most meaningful to track. You know, what actually matters to them day to day. For example, for a condition like Parkinson's, maybe it's the severity of tremors during specific activities, or for Duchenne, like we discussed, their actual top walking speed throughout the day, not just for 6 minutes in a clinic.
Capturing what's relevant to their lived experience.
Exactly. This direct patient input is just incredibly valuable in designing digital endpoints that truly reflect what matters most. And then, from a purely practical standpoint, the importance of device usability, making them easy to wear, easy to charge, easy to understand, and having well-designed compliance strategies, maybe gentle reminders or real-time dashboards showing their own data. That can't be overstated in making sure patients actually use the devices consistently for the whole trial.
That patient perspective is absolutely critical, you're right. After all, if patients aren't willing or able to use these devices consistently, well, the most brilliantly scientifically validated endpoint becomes pretty much useless in practice.
Right.
And it's equally important to consider the perspective of clinicians, the doctors, the nurses, the therapists who will ultimately need to interpret this data and make treatment decisions based on it. Our sources indicate that clinician feedback is, on the whole, largely supportive of integrating digital endpoints into the existing endpoint hierarchies.
Meaning fitting them into the current system.
Yes, treating them as primary, secondary, or exploratory measures, just like traditional assessments are categorized, integrating them seamlessly into that familiar structure of clinical research and practice.
Okay.
And this broad support from clinicians is crucial. It ensures that these digital endpoints aren't just scientifically sound and technologically advanced, but also practical, interpretable, and capable of being integrated into existing healthcare workflows. You know, a new technology, however brilliant it is, won't achieve its full potential if it's not embraced by the healthcare professionals who have to use it.
Makes sense.
Their input helps bridge that gap between cutting-edge research, strict regulatory requirements, and the day-to-day realities of clinical practice. It ensures these new tools become genuinely useful and widely adopted in patient care, becoming a practical part of the medical toolkit, not just a research novelty.
It sounds like a clear win-win then, engaging both patients and clinicians. But have there been any significant pockets of resistance, maybe specific concerns from clinicians that needed to be addressed to achieve this alignment? What anxieties might they have had about moving away from practices they've used for years?
That's a great question. And yes, naturally, there's always a degree of caution, maybe healthy skepticism, when adopting new technologies in medicine. Initial concerns often revolved around things like data overload.
Too much information.
Exactly. How do you integrate and interpret these vast amounts of continuous data without just being completely overwhelmed? Then there's the question of trust. Trust in the device accuracy, the rigor of the validation. Clinicians are trained, rightly so, to rely on well-established methods with long track records.
So how is that trust built?
Well, that's where the V3 framework's emphasis on rigorous analytical and clinical validation, demonstrating equivalence or even superiority to traditional measures, was absolutely key. It provided the evidence needed to build that confidence. Furthermore, practical concerns about workflow integration, how do these new data streams actually fit into their already busy schedules, are significant.
Yeah, that's a real practical barrier.
It is. And this is why involving clinicians early in the design process is so important. Ensuring the data is presented in an actionable, digestible format, not just a raw data dump. And clearly demonstrating the clinical utility, how does this actually help me make better decisions for my patient? It's an ongoing dialogue for sure, but the momentum is clearly towards acceptance and integration.
So ultimately, these advancements, they aren't just about collecting more data or using fancy gadgets. They're really about profoundly improving how we understand and address your health, the listener's health. By involving patients and clinicians so deeply in the design, the validation, the interpretation, we ensure these high-tech measurements are truly patient-centric, reflecting what genuinely impacts daily life, providing insights that truly matter. It's about building trust and ensuring relevance from the ground up for everyone involved.
Okay, looking ahead now, there's a concept that seems to hold immense promise for maybe expanding the reach, the efficiency of clinical trials even further. It's this idea of bring your own device, or a BYOD trial.
BYOD, the holy grail for some, a headache for others?
Right. The appeal is immediately obvious, isn't it? Imagine trial participants using their very own smartwatches, their own smartphones, devices they already own, they're familiar with, comfortable using every single day to collect trial data.
The convenience factor is huge. It really is. This could dramatically widen the geographic reach, the demographic diversity of trials, potentially lower study costs by not having to provide devices to everyone, and just make participation significantly more convenient, less burdensome for a whole range of people. It really sounds like a potential revolution waiting to happen.
The promise is definitely significant, no doubt about it. Offering that kind of unprecedented scale and convenience is very attractive. But, as you hinted, the challenges are equally formidable, especially when you layer on the rigorous demands of regulatory science.
Okay, what are the main hurdles?
Well, while BYOD offers convenience, two big issues immediately jump out: equity and standardization. They become critical issues that demand really careful thought. First, equity. How do you ensure that all potential participants, regardless of their socioeconomic status, their access to the latest technology, can participate equally and fairly?
Right, not everyone has a compatible smartphone or a fancy smartwatch.
Exactly. Not everyone has the same device, or maybe even a compatible device at all. This creates a real potential for a digital divide in trial participation, potentially excluding certain populations, which could bias the trial results.
That's a serious ethical consideration.
It is. And then, beyond access, you have the enormous, really complex technical challenge of data consistency and comparability. If you're accepting data from potentially hundreds or thousands of different devices, you have to somehow account for significant variations between them.
Like what kind of variations?
Think about different operating system versions. An older version of iOS or Android might have different sensor access protocols or processing capabilities than the latest one. Then there's varying sensor fidelity and accuracy between different manufacturers. An Apple Watch's accelerometer might have slightly different specs, different internal algorithms, different validation than a Samsung Galaxy Watch or a Fitbit, even if they're notionally measuring the same thing, like steps or heart rate.
So even the same measurement might not be the same.
It might not be truly comparable without adjustment. And even within the same device model, you can have subtle firmware variations over time that could affect sensor output. So if one participant uses, say, the latest iPhone and another uses an Android phone from 3 years ago, or different brands of smartwatches, how do you ensure the data they collect is genuinely comparable and reliable enough for a clinical trial? A trial that needs to withstand that intense regulatory scrutiny and be the basis for approving a new medicine.
Yeah, that really raises an important question, doesn't it? How do you guarantee the data from maybe 100 different models of smartwatch, each with its own quirks, its own secret sauce algorithms, is truly comparable, reliable enough for a trial that could impact patient lives worldwide?
That is the crux of the BYOD challenge. Yeah. Our sources indicate that the regulatory defensibility, the ability to convince regulators that data is sound, may be significantly lower for pure BYOD trials, unless a very robust strategy for device harmonization and data normalization is put in place and rigorously validated.
Harmonization and normalization, what does that involve?
It could involve several things. Maybe setting strict criteria for which devices are acceptable, perhaps providing participants with a pre-approved list of devices that have been well characterized and meet certain standards. Or, more technically complex, developing highly sophisticated calibration algorithms or data normalization methods. These would aim to scientifically account for and adjust for those inter-device differences, essentially translating data from various sources into a common, standardized language before analysis.
So like a data translation layer?
Kind of, yeah. Without those kinds of stringent controls and innovative analytical solutions, the convenience and scale of BYOD could easily be overshadowed by very legitimate concerns from regulators about data quality, integrity, and comparability. And those things are absolutely non-negotiable for drug approval decisions.
It really highlights this fascinating balancing act, doesn't it? Between the allure of convenience and scale with BYOD and the absolute necessity of rigorous science demanded by regulators. It's clearly not a simple switch. It requires a huge amount of innovation in data science, maybe device manufacturing standards, certainly in regulatory thinking. So what really needs to happen, realistically, for BYOD to become a mainstream, widely accepted practice in clinical trials while still maintaining that crucial scientific rigor?
Well, for BYOD to really achieve mainstream adoption safely and effectively, several things probably need to happen in parallel. First, there needs to be continued, intensive dialogue and collaboration between industry, regulators, academic researchers, tech companies to establish clear, consistent guidelines for what constitutes acceptable BYOD data quality and validation. Maybe industry-wide consortia developing common data models, open-source validation toolkits could help.
Getting everyone aligned on standards.
Exactly. Second, device manufacturers themselves probably need to move towards greater transparency regarding their sensor specifications, their internal data processing algorithms, perhaps even making raw sensor data more accessible in a standardized format for research purposes, though there are commercial sensitivities there, of course.
That could be tricky.
It could. Third, and this is critical, significant investment is needed in advanced data science. We need more sophisticated, validated algorithms for that data normalization and harmonization piece, methods that can robustly account for the inherent heterogeneity across all these different devices.
These are analytical tools.
Definitely. And finally, pilot studies and collecting more real-world evidence will be absolutely essential. We need to demonstrate the feasibility, the reliability, and the regulatory defensibility of various BYOD approaches in practice across different therapeutic areas and populations. It's a compelling vision for the future, sure, but one that still needs quite a bit of significant foundational work to truly realize its full potential while maintaining that non-negotiable scientific rigor. #tag 8. Recap and synthesis.
Wow. We have covered a lot of ground today, really peeling back the layers on digital endpoints in clinical trials. It's clearly a complex area, but also incredibly exciting, isn't it? This frontier that genuinely promises to redefine how we measure health. So just to bring it all together, let's quickly revisit some of those key regulatory insights we've discussed. Using SV95C as our sort of guiding example, let's reinforce what truly makes a digital endpoint regulatory grade. Think of these as the essential lessons learned from this whole transformative journey.
Absolutely. A quick synthesis. Think of this as our mental summary table, highlighting the crucial elements regulators are increasingly looking for. First, regarding endpoint type. The big takeaway here is that hybrid endpoints, the ones combining features of both a clinical outcome assessment and a biomarker, they're definitely valid and increasingly seen as powerful. But, crucially, they need that dual evidence. You have to demonstrate both functional or symptomatic relevance to the patient and physiological significance. SV95 nails this by measuring both real-world function and a physiological capacity.
Right. Then there's that critical aspect of analytical accuracy. The real revelation with SV95C was achieving that lab-grade precision, remember. Less than 2.5% error compared to gold standard motion capture. That proved the V3 framework, especially the analytical validation stage, really works. It showed we can get almost perfect fidelity from a tiny device worn on the ankle out in the messy real world. That level of precision is why V3 is so powerful.
And that ties directly into reliability. That interclass correlation, the ICC, of around 0.97 over months for SV95C, that's an absolutely essential benchmark. This incredibly high reliability is required for comparing measurements over time, what we call inter-period comparison. It means you can trust that changes you observe over months are real changes in the patient's condition, not just random measurement fluctuations or the device behaving differently.
And what really stood out for me was the responsiveness of SV95C. Detecting that functional decline in DMD patients in just 3 months compared to 9 or 12 months for the traditional walk test. That dramatically enables earlier decision-making in trials, faster interventions, quicker stops for ineffective treatments. Ultimately getting effective treatments to patients significantly faster, it changes the whole timeline.
And that responsiveness is amplified when you consider the patient-reported relevance. Our sources showed over 70% of patients actually prefer wearable monitoring in some studies. This high acceptance, this preference, it deeply supports patient-centric endpoint validation. It ensures that what we measure truly matters to the individual's experience and makes the data collection sustainable because patients are on board.
And to make those precise measurements truly meaningful, we talked about the critical role of anchor-based thresholds. For SV95C, remember that change of nadical 0.10 to nadical 0.20 meters per second was identified as clinically meaningful. This really demonstrates the preference now for combining those anchor-based methods, linking to patient experience and other scales, with distribution-based methods, statistical properties, to truly translate a numerical change into a real-world impact that resonates with both patients and clinicians. It's about defining what matters.
There's the practical but absolutely paramount challenge of the missing data strategy. We learned that for digital endpoints, you need a robust, pre-specified approach, using things like functional imputation, but critically, performing rigorous sensitivity testing. This means you must explicitly address the risk of data being missing. Not at random MNAR, remember, where the missingness is linked to the condition and you have to meticulously document your entire strategy in the statistical analysis plan. That's essential for ensuring data integrity despite the complexities of real-world data collection.
Got it. And finally, we highlighted that foundational importance of terminology alignment. Developing and using a harmonized glossary like the EFPIA lexicon isn't just bureaucratic box ticking, it's crucial for streamlining the entire process. It prevents misclassification of these digital tools, ensures clear, consistent communication among everyone involved, sponsors, regulators, CROs, researchers, and that ultimately accelerates the regulatory review process, helping get effective drugs to market faster.
And here's where it gets really interesting, I think. When you bring all these diverse elements together, right, from the cutting-edge technological precision of the devices themselves to the deep insights you get from direct patient experience, and the foresight and willingness shown by regulatory bodies like the EMA, you build this undeniably robust, compelling case for digital endpoints. It's this comprehensive, multifaceted approach on the right, tech, patient, regulator, data science that is fundamentally changing how we measure health outcomes. We're moving beyond those traditional sporadic snapshots towards a continuous, precise, and profoundly patient-relevant understanding of health and disease. It really feels like a new era for medical research. #outro for thing.
And that brings us to the end of this deep dive. We certainly journeyed through a lot, the rise of digital health technologies, that landmark approval of SV95C, dissecting the V3 framework, grappling with data challenges, and peering into the future with BYOD. Hopefully you feel like you've taken a fast track to being pretty well informed on what is truly one of the most transformative areas in medical research today.
I think the profound impact of these advancements on drug development, on how clinical trials are run, and most importantly, on the future of patient care, really can't be overstated. We are genuinely moving towards a future where our medical understanding isn't limited by those intermittent clinic visits anymore, but is enriched by continuous real-world data that truly captures the dynamic, the nuanced nature of health and illness right as it happens in everyday life.
So as you go about your day, maybe glancing at your own smartwatch or just with your smartphone in your pocket, here's a final thought to maybe mull over. How might this ability to continuously, precisely, and meaningfully measure your own health right from your wrist or pocket, how might that redefine what it even means to be healthy? And how might it change how we interact with our own well-being and with the healthcare system in the future? What new insights, what new possibilities might emerge when your personal data becomes a dynamic, living health